The scientific method is a thorough, step-by-step process used to investigate natural phenomena, gather knowledge, and develop reliable conclusions. It involves several key stages, each designed to ensure that the conclusions drawn are supported by evidence and can be verified through repeated experimentation. Here's a detailed breakdown of the process. Number 1. Observation. The process begins with observation. This could be anything unusual or interesting in the natural world that sparks curiosity, whether it's the behavior of animals, a physical phenomenon, or a pattern in data. This observation leads to the formulation of a question that needs an answer. Example: you notice that plants grow faster in some areas of your garden than in others. The observation leads to the question: why do plants grow faster in certain parts of the garden? Number 2. Research. Before forming a hypothesis, it's crucial to gather background information. This involves reviewing what is already known about the topic by looking at scientific literature, studies, or prior experiments. This step ensures that the question being asked hasn't already been thoroughly answered and helps refine the scope of investigation. Example: you research factors that affect plant growth, such as sunlight, water, soil nutrients, and temperature. Number 3. Hypothesis. A hypothesis is a testable, educated guess that provides a possible answer to the question. It's a predictive statement that can be supported or refuted through experimentation. Importantly, the hypothesis must be testable and falsifiable, capable of being proven wrong. Example: based on your research, you hypothesize, plants grow faster in parts of the garden with more sunlight. Number 4. Experiment. The next step is to design and conduct an experiment that will test the hypothesis. An experiment must have a clear procedure, controls, and variables. The independent variable is the factor you change, for example, the amount of sunlight, while the dependent variable is the factor you measure, for example, the growth rate of the plants. All other conditions should be kept constant to ensure accurate results. Control group: this is the group that remains unaffected by the independent variable, serving as a baseline for comparison. Experimental group: this group experiences the change or manipulation of the independent variable. Example: you set up two groups of plants, one in full sunlight and another in partial shade, both receiving the same amount of water and nutrients. Over a few weeks, you measure their growth. Number 5. Data collection. During the experiment, you collect data. The data can be qualitative, descriptive, or quantitative, numerical. Proper documentation of observations, measurements, and any irregularities is crucial to maintain accuracy. Example: you measure how tall each plant grows every few days, keeping a log of growth rates for both the sunlight and shaded groups. Number 6. Analysis. Once the data is collected, it needs to be analyzed. This involves comparing the results between the control and experimental groups, using statistical methods if necessary. The goal is to determine whether the data supports or contradicts the hypothesis. Example: you compare the average growth rates of plants in full sunlight and in partial shade. If the sunlight group consistently grows faster, it supports your hypothesis. Number 7. Conclusion. Based on the analysis, you draw a conclusion. If the data supports the hypothesis, it may be accepted, but this doesn't prove the hypothesis. Further testing by others is required for validation. If the data contradicts the hypothesis, it is either rejected or modified. In either case, new questions may arise, leading to additional experiments. Example: you conclude that plants exposed to more sunlight do grow faster, which supports your hypothesis. Number 8. Communication. Once a conclusion is reached, the findings are communicated to the scientific community. This can be done through publishing in scientific journals, presenting at conferences, or sharing results in other ways. Clear communication allows other scientists to review the methods, replicate the experiment, and further validate or challenge the results. Example: you write a report on your experiment and submit it to a gardening journal, where other researchers can review your findings. Number 9. Replication. Replication is a key aspect of the scientific method. Other scientists should be able to replicate the experiment under the same conditions and obtain similar results. Replication confirms the reliability of the original findings and helps build a stronger body of scientific knowledge. This structured process ensures that scientific investigations are objective, reliable, and based on evidence, allowing us to better understand and explain the world around us.